This presentation is about A-kinase anchoring proteins, and it's by Jerome, Chiklokozo, and Samito. Introduction. The A-kinase anchoring protein, AKAPs, are a group of structurally diverse proteins which have the common function of binding to the regulatory subunit of protein kinase, A, also known as PK, and confining the holoenzyme to discrete locations within the cell. A variety of gel overlay, interaction cloning, and proteomic approaches have identified up to 25 unique AKAPs, some of which are members of the complex gene families with numerous splice variants and isoforms. With many kinase and phosphatase genes that may have been identified, the research community now is focusing on understanding how these enzymes orchestrate phosphorylation events inside the cell. AKPs also bring together signal transduction and signal termination molecules in the convergence of signaling pathways. Structure. The structure of AKP250, also known as gravin or AKP12, based on analysis of milligram quantities of recombinant proteins expressed in E. coli, suggests that AKP is probably an unordered scaffold acting as a necklace on which jewels of structure function, e.g. the RII binding domain that provides docking sites on which signaling components can be assembled. Recent results suggest that AKP250 provides not only a toolbox for assembling signaling elements, but may indeed provide a basis of spatial constraint observed for many signaling paradigms. The spatial dimension of the integration of cell signaling will probably reflect many functions performed by members of the AKAP.
Well, the AKA anchoring protein really is targeting sequence, as I've already said. The R1 and R2 interact with apathetic helix on the AKAP. Anchoring kinase facilitates the precise activation of the PKA catalytic subunit following elevation of the second messenger, which is the CAMP. These anchoring proteins bring together kinase, phosphatase, phosphodiesterase receptors, and in channels is important for signaling pathways at three distinct sites within the cell: the centrosome, the Golgi, and the plasma membrane. By assembling various signaling partners, these proteins are able to modulate cells, progress membrane trafficking, and channel function. A good example of how AKAPs work, I chose the coupling of the electric signal to construct how this works. Well, this is the heart. You know what the heart does? Pumps blood to other parts of the body for oxygen. We break it down to coupling. The coupling of this electric signal to construct in the cardiomyocytes is initiated with the calcium influx 2L type channels, which activate intercellular calcium release through the radion receptors located in the sacroplasmic reticulum, and thus induces a global increase in calcium that triggers contraction. The EC coupling is a process whereby an action potential basically triggers the myocyte to contract. The arteria and the ventricular myocyte is activated by action potential synuclein node in the walls of the right atrium, often referred to as the pacemaker. Well, basically, for the purposes of coupling of the EC to go through, it goes through three phases. Phase 1, A, B, and C. What happens in A? Well, as you can see in A, the protein AKAP79/150, and the short form of the AKAP115/18, which is an alpha and beta, maintain signaling complexes tied to the L type calcium channel to enable calcium influx. When the runner receptors at the SR are bound to the M signaling complexes, as it is responds to the increase of cytosolic calcium. The messenger AKAP clusters PKA, PDE4D3, and PP2A to regulate the phosphorylation status of the RNase receptors and the resultant calcium to releases from the SR. In 3, the last one, transport of calcium back into the SR via the sacroplasmic reticulum or endoplasmic reticulum pumps 2 is regulated by phosphoblum. The AKAP long inform bring pKa into complex with phosphoBAM and CER. While oil or the AKAP complex contribute to each phase of the EC by coupling, optimizing the phosphorylation of ion channels and construction of proteins.
Okay, on this slide we are talking about the important discovery of AKAP. A kinase anchoring protein influence level of glucose in the body. The team of researchers led by Simon and John Scott revealed for the first time that AKAPs influence the level of glucose in the body by coordinating the special positioning of phosphate neutrally occurring enzyme that can counteract the effects of kinase enzyme. The observation that AKP150 functioned by coordinating phosphate activity in the cell reveals a new role of these anchoring proteins in the control of the glucose metabolism and related to metabolic disorders. It also suggests that new drugs that interferes with the role of anchoring proteins are possible therapeutic intervention to treat chronic disease such as diabetes. The researchers use imaging techniques as well as genetic modification of isolated insulin secreting cells and whole mice to investigate the impact of anchoring protein on glucose metabolism and insulin release. Mice that lacked the genes for the AKP150 anchoring protein produce less insulin from the beta cells in the islet of Langerhans. However, they copied better with limited amounts of hormones due to the increased sensitivity to insulin in the target tissues. The scientists showed that these effects are due to the 7 amino acid sequence in the anchor protein that directly interacts with the surface of the phosphate enzyme. Conclusion. A kinase anchoring proteins are a group of structural device proteins, and its complex contribute to each phase of EC copying by optimizing the phosphorylation ion channels and contracting proteins. Intercellular targeting of AKP served to position distinct signaling complex on a given organelle. Thank you.